anything we sample. But temperature does, and temperature changes a lot by about 20 degrees C, 25 degrees C sometimes. Yeah, yeah, I guess you could say going from 2,000 meters up to the surface is kind of like driving from, you know, Vermont down to Miami in in like a couple hours. It's a really fast temperature change for these deep sea animals. So we try and keep them closed and sealed boxes as much as possible so we can prevent them from stressing out.
Are the bio boxes insulated?
They are insulated, yep, when they seal properly.
Wondering, would you need to do like a formal transect to get a full like habitat assessment or species distribution?
Yeah, that would be helpful. So it depends on how you set up your sampling plan. These are exploratory dives. We don't know what we're going to find. We don't want to really limit ourselves to only what we see in a, a 2 meter by 100 meter transect. So we want to kind of meander and get a handle on what's in the area. And you can sometimes miss things when you constrain yourself. But yes, you do miss the ability to compare apples to apples when you don't do transect type exploration.
Yeah, someone actually asked earlier, and this kind of ties in. I can find it. I can't find it, but it was essentially, are we doing conservation or research? And we're doing exploration. Is that
right?
What's the kind of Venn diagram of those in your all's mind?
You could argue that exploration is a precursor to research, but research can also be exploration and vice versa.
Right, not all research is hypothesis driven. Sometimes you just gotta go see what's out there and see what the world is revealing about itself.
Yep, and these observations will yield insights that we can use to put into proposals and get potentially more support to do more intensive research projects that try to test the questions that we've developed based on our initial observations.
Yep.
Jake, is Argus back to the regular HD again? I don't think you're on us.
I can answer that. We're Argus has a mini Zeus camera, so it's a spare HD camera that we normally have on our other vehicle at Atlanta. So that's what we're using.
Well, thanks, Renato.
Sure. He was off SPL, so I thought.
Oh yeah, sorry, I wasn't listening.
What?
But Jeff, do you know, we were kind of discussing it. Were there any differences between those two cameras?
For the viewer at home, no, not really.
Yeah.
For us, there's some differences. We don't have quite as many controls over it. Yeah, otherwise it makes pretty HD pictures. It keeps a nice eye on Hercules.
Much better than the
black and white security camera last night. Yeah.
It was
Just
kind of
bit.
funny though. It looked like it was like watching Herc through like a filter of a 90s show or something.
Oh, I thought it would have been great to have filmed scratch
Yeah.
type things through it like an old film reel. But and
Silent
here, yes, in the 1920s when we used to dive here.
All right, the mezzo straightened itself out.
Nice. How about the lights?
No, not the lights.
Okay. Good. I'm happy about the... Oh, look at that. It's too slow. Come on. Come on. Where's the speed? Too fast. Oh, look, they're almost synced up.
Oh, lovely.
Yeah, I can.
Jake, you want to do another look at the gauges there? Quick?
No, I don't think they can be because they're two different ranges. What if I... Let's see. Oh no, I hit stop. I hit the wrong button. Yeah, I'm just messing around with it. Listen to Jeff too much over there. Is that how fast it is? Is it like that? That's probably fine. Yeah. We have a cursor-shaped target up ahead. It's an arrow.
Rennie, is the color choice for Argus tracking tonight a deeper blue to be a better contrast to the underlying map, or some other reason?
Probably. These are usually the colors that we go with. I think lately we've been doing a light blue because of the colors on the map. But yeah, I think it looks better. I think Aaron might have set that up. Or maybe it just was default and wasn't changed. If you have a preference, we could switch it up.
No, I don't have a preference.
Yeah.
This is great.
Steve and Beth, for the Niskin water samples, are you also potentially measuring parameters like oxygen, pH, or trace metals?
So there are sensors on Hercules that are continuously measuring temperature, conductivity, salinity, and oxygen concentrations. And actually you can see that data if you go to the Nautilus Live webpage in the little sidebar next to the video choices. You can click on more data and see some of that data. We do not have a pH sensor on the vehicle. For these samples that are coming up, we're mainly collecting them for eDNA, as well as for some of the samples, I filter them for microbial analyses, and I do take a small subsample for measuring nutrient concentrations, because that's a parameter that I'm interested in, but I am not doing sampling for trace metals. That requires a whole nother level of setup that was beyond the scope of what I could do as one person. The next expedition that's coming out to the waters around Hawaii, the Chautauqua Seamount exploration, will have a trace metal component. So yeah, if that's something of interest to you, definitely check out the next expedition. But yeah, oxygen is definitely something I've been interested in and looking at the data in real time as we've been diving, because it's an important parameter for thinking about where the animal communities are, as well as how mineral crust might form on some of these materials. None of our sampling got into the oxygen minimum zone where the oxygen concentrations can get really low in the water column. We were always they're slightly below it or well below it, but it is an interesting parameter nonetheless. And we could definitely see some changes as we would move up slope that the oxygen concentrations in some cases would decrease. Yeah, so that'll be something that we look at.
That's great. I have lost access to comments again temporarily. So
Okay.
Thanks for your comments and questions.
Yeah,
I'll be back with you shortly.
Yeah, and keep sending.
Keep sending them in. They'll just do a backlog here.
Yeah, we've had some.
Don't at me.
Some satellite connection issues or something going on today. All the internet things are wonky.
Gremlins in the system.
Yep. Yeah, and if anybody who's listening right now was part of some of the planned ship-to-shore interactions that were scheduled for today, I know at least some of them had some connectivity challenges, so apologies for that, and hopefully we can get those rescheduled. But such is the nature of being on a vessel in the middle of the ocean, trying to send all of our data up to space, satellites, to get it back to you on land, doesn't always work seamlessly.
Yeah, since I'm not really able to see comments consistently, I would encourage you to check out some of the new content that went up around this expedition, especially for those who have been following and would like to see some of the highlights. There's a video called Tretipleural Sponge of Unnamed Seamount A in the Wentworth Seamount Chain. Another video that is up right now is the Bamboo Coral of Unnamed Seamount A in the Wentworth Seamount Chain. And then we have a photo album and a blog. The photo album is called A Tale as Old as Time, A Geologic Love Story. And the blog is the EV Nautilus Field Guide to Deep Sea Geological Features. So some great fun content inspired by our exploration this past 2 weeks.
Yeah, we might want to update some of the titles of those because we actually don't know if this is an extension of the Wentworth chain. That's part of the hypothesis of this expedition, is to verify that that is actually true, that this is the southern extent of the Wentworth chain, which is to the north. So we don't want to get ahead of ourselves and commit it because we don't know yet if it's actually part of that chain.
That's a great point. I will definitely pass that along to our social media team.
Yeah, and we've definitely come across some surprises on this expedition in terms of the rocks that we've collected. You know, I kind of came out here with an assumption that if these seamounts are super old, then they should have super thick manganese crusts on them, and they haven't been as thick as I was anticipating. Doesn't necessarily mean that they're not old, just means that my assumption of linear crust formation over time is maybe not accurate. We've also seen some interesting rock types with kind of consolidated sediment, sandstone, strange things, what we were not necessarily expecting. So the seamount chain, if it is a chain, is actually more complicated than we might have anticipated, which is exciting to try to unravel that story using all the different techniques that we have at our disposal.
Yeah. Another kind of factor of that, the complication there has been kind of the currents and trying to kind of predict where currents are coming and moving. And somebody's wondering if we're going to try to model current activity based on observations and recordings from the dives.
I don't know if we could model it. I think probably just in the sense that I don't know that we like rigorously did enough observations to try to have enough robust data to model something like that. But we can at least, I could imagine as someone goes through the video annotations of marking, looks like the flow is coming from this direction in this orientation on this seamount to kind of get a general sense. But we also have to be mindful that every dive we did was one snapshot in time. And so that doesn't mean that the current always moves in that direction. And we always did, you know, one dive at one place, another dive at another place. So we never saw that kind of temporal variability, which would be an important part of trying to model currents.
There are some publicly available data sets, hydrographic data sets for the deep ocean, but they're at relatively coarse resolutions. So you might have data points that are on the scale of kilometers or tens of kilometers. And that could be used and plugged in as a parameter in a model, but it doesn't give you that fine detail that we see on the seafloor with our own eyes. It doesn't tell you one ridge, you know, is going to have a garden of precious corals and the other ridge is going to have a garden of bamboo, black corals. So that's kind of data you need to have higher resolution information for, which can usually be collected by ship-based platforms or drifters or something like that. So it needs to be measured much more robustly. But a few of the parameters that might go into modeling that we did collect are high quality multi-beam bathymetry, and all the variables you can pull out of a multi-beam bathymetry is very important. We collected, and we will have soon, once it's analyzed, presence and absence data, which is usually the first step for determining habitat suitability or conditional models for, you know, where corals and sponges might want to live. We have species, or we will have species identification data coming from collections, for example. And then we have all the other parameters that life might find sensitivity to, like temperature, salinity, oxygen. Yep. So those, you know, we're slowly building this data set.
Thanks, Dave. Yeah, so we'll be starting the dive at around 1,960 meters deep, and we'll be moving kind of gently up slope. I think the target end depth is about 1,600 meters, something like that.
Yeah, although if past dives have been a guide to the future, often we surpass our goal and spend a little bit of time exploring some other things. I don't think we'll get any shallower because we are definitely targeting the summit of this feature. But we might go up and down. And I think we're about 2/3 of the way there.
Yeah, we got 25 minutes left in our descent. Probably 20 minutes.
Okay.
Would it be possible to see the bathymetry of this seamount?
Sure. Let me just double check. Aaron might have set it up here. Yeah. So on MB Proc, if you put that on.
If you press the...
It's up.
Oh, thank you. So this is the seamount
We've got some paparazzi in here.
we're diving on. And as Beth said, we're aiming, our dive track is heading towards the summit.
Yeah, great.
So you can see here, we're kind of on this north ridge.
Yeah. That's a very pronounced ridge. Could that at all be due to the multi-beam collection, or do we think that's real?
That's real.
Okay, very cool.
Yeah, because it was in
It's like a target.
multiple different directions. Yeah, we see these a lot. I shouldn't say that.
It almost looks like a compass rose.
Yeah, some seamounts have very prominent ridge features that are kind of
Oh my gosh, it does look like a compass rose.
Uh-huh. Very Nautilus-like.
Very
Yeah.
Nautilus. What a good one to end on. I'm going to copy Chris Kelly here and take a watch photo. Hey guys, back row, look here. Steve, where are you?
Oh. What?
Steve, get in the photo.
Oh.
Nice.
Yeah.
Yeah.
You're a fun guy. Oh, I know. Yeah, I did. That's my response to your joke was took a long.
Do you, by chance, have a slope layer on that, Pathy?
I know I have it in, I think I have it in HiPAC. I don't know if I have it in here. I mean, I could make one real quick. Let me see. I think I have it in HiPAC. Oh my God. Yeah, it's pretty rough, Steve. I think the contours are going to help you better, but you know, on HiPAC, there's the slope map.
Yep. Okay. Is there a color ramp?
Yeah,
Approximate?
I think that might be. I might have a PNG of that. This computer is freezing. Not a good sign.
Don't worry about it if it's too complicated.
It's fine. Just disconcerted. Cool. All the icons are gone on my start bar. I blame you, Steve.
What is going on here? So this is our last chance to come up with a team name.
We've come up with so many team names.
I know.
Which one is the final?
Been defined by many identities.
I don't know how we're going to choose.
My vote's for intertentacular sclerites. Just keeping that on the forefront.
I don't remember ever hearing that
I
one,
don't
so I think
either.
it's too late to be entering new.
No, this was.
I
I think that was when Chris was.
Yeah, Chris sat in.
Chris
Oh,
sat
that's
in.
right, that's right,
Oh.
yeah.
So I guess we can't be because Beth wasn't here. when we came
Yeah,
up with
right.
it.
It was not. I was not
No, but
part
we can
of
use
that.
highly altered. I think that's highly
That's right.
altered something.
Highly altered tentacular sclerite.
Highly altered.
I want to go with duty and leisure.
Duty and leisure.
Go back to the first one. Yeah.
I'm liking duty or leisure.
Duty and leisure.
Has anyone written these down yet, or?
We don't need to. We remember them.
I went down to the lounge. and
D&O.
They also had microbes in the front, sponges on the side.
Right.
That's right.
Oh,
That's right.
Actually, I like highly altered microbes. That's kind of neat.
Ham.
Ham.
Hey, well, it's actually
could be like-
could be true. We
could
are
be like
highly
us,
altered
yeah.
microbes.
Highly
Yeah.
altered.
Mm.
And we're hams.
Yeah.
Hams.
Kind of works.
Highly altered microbe. The
Oh.
acronym ham.
Oh, it's got an acronym.
Ham
watch.
hams.
Ham watch.
Yeah.
also very reflective of my post-watch
Ham.
ritual.
Yes.
Post-watch snacking.
Ham fridge.
Ham fridge. Oh. Do we want to be associated with that ham
Ham,
fridge, though,
ham
guys?
fridge.
Kimchi bowls.
We had this long running joke about the ham fridge. The fridge, like you open it up, there's like cold cuts in there. It smells like ham. So we were on watch, and we had this thing where you would try to pass off that you were from Hamfridge, Massachusetts.
Okay. Yeah.
But there was someone from New England who wasn't in on the joke, and they were like, "Now where is that?"
Oh no.
And I had
Oh
to
no.
like talk my way out of it. Yeah, Hamfridge, Mass.
I've never heard of Hamfridge.
Ham fridge. It's awesome.
Oh.
How many more minutes till we reach the bottom?
13
Faster,
guys.
yeah. It's going downhill fast.
Yep.
Yeah.
We wouldn't want to disappoint our audience. This is what they come for.
Yeah.
Yeah, I thought we were the prime time watch, but it turns out we're probably prime time for Hawaii and kind of like the silly watch for folks on
We're
the
the
East
late
Coast.
night. We're
Yeah.
the
Yeah, late night
Yeah.
entertainment.
Yeah.
Which is when all the best talk shows come on.
Right.
That's right.
Although we are the morning show for the East Pacific or the Western Pacific Rim.
Oh, yeah.
So
We're like, yeah, the drive time.
Yep.
Getting to work.
Yep. talk
Okay.
radio, morning show,
Who's got the weather report?
which are quite silly too, someplace.
Yeah, the science portal.
Did we consider highly altered tunicate hunters? That just came
Oh.
through.
Oh.
It's not much of a sport.
Wow.
Also not a cool acronym.
Oh, I should.
Okay, here
I
we
would
go.
not be proud of being a tunicate hunter.
No.
Okay, here we are.
What about crab hunter? Would that be better? We were
Yeah.
looking for those crabs for a while.
That's right.
Yeah, we were. That's true. Or it could sub for coral hunter.
The acronym also doesn't work out terribly well to write.
No.
Yeah,
The altered coral hunter.
sounds like a clearing your throat.
Very guttural, yeah.
I'm going to try and do a regression analysis at the end of the season, see if I can identify if the bad jokes are consistent with loss of viewership.
Oh, yeah, there you go.
I don't think
Wow.
it's loss. I think it's a gain, really.
Yeah.
We got
I would
a
too.
ton of really positive comments at the end of our last shift. Like, we rock.
Yeah.
That was all blue water, wasn't it?
Yeah, exactly.
Yeah, it's
right?
3.5 hours of blue water.
We still had people
Those
watching.
are sympathy comments.
That's impressive. Yeah.
Right, it's all of our parents.
Hi, Mom.
Yeah, thanks for watching, Mom.
Yeah, we've had pet viewers too. It's quite common.
Pet viewers?
Pet viewers, yeah.
What is that?
I don't
remember-
understand.
Like a dog?
Yeah, a dog. A dog used to write in.
Someone
Oh.
had a parrot
remember
that used to
this?
say nav bridge. That was a long time.
There used to be a
watching.
member of the ROV team
we
whose
gotta get
pet
to the
used to write
bottom.
in.
Bridge nav. Bridge nav. Bridge nav. Bridge nav.
Paul's probably up there listening to SPL going, what?
Yeah.
George would be going, what?
Yeah.
It's true.
I was distracted. Did you all ask for suggestions of acronyms that work with ham?
Say it again.
Did you all ask for more ham?
Did we
Names?
ask for more ham?
Is that what the internet's providing?
Yes.
Yeah,
did not.
well, if we're going with the ham as an acronym and we're not happy with highly altered microbe, which I think is great.
I think it's great too.
Yeah, if
Yeah.
our audience wants to send other acronym
We have
fillers.
humans autonomously mapping,
Okay.
Oh.
highly arranged mollusks,
Oh.
happily authoring
Ouch.
memorandums.
Oh,
Okay.
never.
Never.
I like the creativity, but I don't like the sentiment.
I like the fact that they're playing along.
Yeah, no, this is great. Yeah, ham it up with us.
Ooh.
Oh, that was very good.
Yeah, we could just make a shift T-shirt with all of these options for our shift name.
Yeah.
Oh my God.
Or you guys know those like magnetic tile things that you can have on your refrigerator that like have a bunch of
Yeah.
words that you can organize.
Oh, yeah.
We can like make a little custom set, all HAMM words.
It's a very small market for that today.
Oh, and I've lost the comments. Sorry, y'all.
That's not
I think
losing
maybe the
comments.
comments
They
just shut
they just stopped
themselves down.
writing
Like, this is going off the rails. All right. We got about 300 more meters left We're on the bottom,
Yeah.
which is good. I don't know if we can handle much more depth.
I did see right before this crashed again, though, someone said, wait, if these are not part of the Wentworth seamouth chains, then where did they come from?
Yeah, this is a great question. Thanks for bringing us back to science. If you look at a map of this part of the world that includes seafloor features, right, you'd see that the Hawaiian island chain is kind of a west-northwest to east-southeast kind of trajectory of islands and seamounts until it gets to the Emperor Seamount chain, kind of makes a little dogleg up to the northwest. But if you look closely, there's a whole bunch of other seamounts that kind of cross obliquely to the northern Hawaiian island chain. Through work with like dredging, collecting rocks to try to date them, it looks like some of those seamounts are older than the northwest Hawaiian island chain. Both to the west of us, as well as in other places around the plate in this area. And so the question is, that's actually a question we're trying to figure out, is like, how did these seamounts form? Because it's unlikely that they formed over the same hotspot that formed the the Hawaiian island chain because of their orientation. And so that's why we're here, actually, is to try to figure out the ages of these seamounts and the composition of the seamounts to try to then back calculate where did they form and how long ago, to then understand the plate movement over hotspots. Yeah, so still to be determined what the origin of these environments, where did they come from?
Cool, thanks.
And I don't know, Steve, can you help me out? Is the cruise plan developed for the expeditions? Is that publicly available somewhere? Or any of the like recordings of the plan for the expedition that we give these like public presentations?
So I think there's an
overview.
expedition overview at the beginning of the season.
Yeah,
that
right.
comes out. Yeah. That is sent to the scientist ashore community.
Okay.
I know that. I don't know how publicly available it is. I do know that the website has basically the same kind of language on pages for each expedition that it's upcoming in the season.
Yeah.
So I don't know if it's a single document, but it's on the website.
Okay.
Yeah.
Well, we can try to hunt that down. But in the cruise plan for this expedition, there's some background about that kind of geological context for what we're trying to do, with some references to the existing data about the ages of seamounts in this region. So if our audience is interested.
Yeah, someone is. Can you describe again how the orientation of these seamounts that we're on differ from the Hawaiian chain?
Yeah, so if you were to look, for instance, at a map of the seafloor, the seamounts that we've been diving on in this expedition have a more northwest to southeast lineation than the northern Hawaiian chain, which is more west-northwest to east-southeast. So they kind of cross each other, not at a 90-degree angle, but at an oblique angle. And so it looks like we've got this line of seamounts on the south side of the Hawaiian island chain. And the thought is that if you were to connect the dots, there's a string of seamounts going off to the west-northwest. That is the Wentworth chain. And so we think there's a hypothesis that these seamounts we're diving on are the southern extent of that Wentworth chain. So it kind of crosses over the northwest Hawaiian Islands.
Yeah, Google Earth has a great way to zoom in on those. You can actually see at least the preliminary. They're not as well mapped as they are after our cruise.
Yep.
But you can kind of see the general chain there.
Yeah, and I realized I was making all these hand motions in space, and Channel 3 is not looking at us. So
it was really
that was for
helpful,
your
though,
benefit.
For me. Thank you.
Yes.
I can fix that.
Yeah, no worries.
I think the moment's gone.
There you go.
Oh, yeah, no, not right. No, thank you. Front row, any sign of bottom in your sensors up there?
Yeah, under 100 meters.
All right, great. Is everybody ready for holiday cheer once we get back to port in a couple days? I feel like I've been existing in a holiday vacuum out here.
Yep.
No, I'm definitely not prepared for the holidays.
I think
Still
I'm
have
still in
shopping to do.
denial a little bit.
Yeah,
That I'm going to have to do all my holiday shopping in about 24 hours
yeah, right.
before it happens.
I think my wife has already made
I'll
and
stop.
hid the Christmas cookies. So
It's before coming out here, I was messing around with my external hard drive that has all my music on it and didn't think to put on my holiday music. So you're
Oh, yeah.
welcome.
Mm. Yeah, I am wondering when I'll hear the first song out here. Who's going to do it? It's kind of surreal going from, you know, this kind of thing we do kind of feels the same year round, and then you just go back to shore and everyone's, celebrating the holidays. It's,
Yeah, it's like
there's no
anti-time out here.
seasonality, yeah, to this because we're in a dark box
155.
that's constantly air conditioned and same temperature year round.
Jeff, it looks like you've put up the Argus sensor for seeing the bottom in channel 3. Jake, can you explain what the audience is looking at?
Yeah, that's our sub-bottom profiler. So the top, well, it's also there's a sonar in the top half of the screen. That's one of our Herc's sonars. And then the bottom half is the sub-bottom profiler on Argus. The top portion of that is our high frequency output, and then the bottom is the low frequency, so that's what penetrates through the seafloor. So we use the high frequency part as an indicator of when we're seeing the
You
bottom.
can see bottom and bubble.
Okay. Thanks, Jake.
Yep.
And it looks like you guys have now set up Hercules below Argus, which we can see in channel 2. And it looks like bottom maybe coming into sight in Herc's cam.
Pretty snowy here.
Yeah, really turbid.
Tis the season.
It's going to be you, Jake, isn't it?
Yep.
The songs.
You can just bust out into song if you want to.
there it is.
Just make sure you're muted.
In high school, I actually was in chorus, and we sang the whole Frozen soundtrack. It was fantastic.
Hey.
Wow. I did
Yeah.
not know you were that young.
I'm here for
I
that.
am very young.
I am very young. You have no idea how young I am. How young
awesome.
I could be.
I realized as I said that, I was like, wow.
It's aggressive.
So in all the 13 dives, you want to share that with us now? And all that blue at a time to hear your beautiful
We're going to
voice.
switch you over to DVL.
Getting our first view here of Ho'oikaika Seamount.
Quick DVL reset. There you go. We'll do a better one when we're there.
Yeah, really turbid water here. Well, doesn't look like this dive is going to disappoint already so far. Many different
All
species.
Yeah, at least, yeah, you might have 8 to 10
At least,
in this area.
yeah.
Maybe more, actually.
Kalamehu, can you remind us what Ho'oikaika means as a name?
Yep. So Ho'oikaika is basically to like strengthen. I don't know what the genealogy or the intent was in the naming, but Ho'o is basically like is like to make something happen, and then Ikaika is just being like strong. So I don't know. The way I think about it is Ho'oikaika is basically just make strong.
Make strong.
Yep.
All right.
Acro, did we want to do our permission?
Yes, we do. Are you all set?
Yeah, we're all set up here now.
Okay.
Great, thanks. So we're going to be going radio silent here to conduct a private ceremonial protocol that acknowledges and honors the biocultural resources that are clearly present and that we'll be interacting with and impacting here on this dive. So we will be back with you shortly. And at this time, I invite everyone in the control van to go ahead and mute your microphones. That was our final protocol.
For this shift, well, we might have one more shift if we're doing 16 hours.
Okay.
Yeah, if we do 16 hours, we're doing the recovery
Yeah,
too.
so might be the last time, but it may not be. All right.
Some balancing here at your convenience.
What are we
Yeah, sure thing. Let's do it.
Rennie, can we talk ship's move?
Sure. We will be, as long as we want to head on top of this ridge, we'll be going
Jake, can you get the
155.
lasers off?
Yes.
Does that make sense with what you guys are thinking back there?
Let's give it a try.
Hey,
Sure. And once they get done with this, if you want, we can just peruse around the area
Alright,
before
yeah,
we
for
get
sure.
moving. It's
is
up
that
to you.
is that our normal? Yeah, I guess it is.
Steve, suggestions?
I don't
Should
even
we
think we have to cruise. We can just pan
Alright,
around.
Back
the-
balance first.
say it again?
I don't think we have to cruise. We can just pan around a little bit. There's enough to look
Okay,
at
yeah.
exactly where we are.
Yeah, so maybe let's pause on putting in that ship's move, Rennie.
Okay. I'll holster that.
That
Be
way
ready
we
to go.
won't have to stop as much.
Yeah.
Also, if we get a handle on what's here first.
Sure.
All right, we're good. Thank you. Oh, that's white.
There's just tons of stuff. I don't think I've seen that spiky coral off on the right-hand side behind the minute.
Oh,
A couple of primnoids on the right-hand side. I saw some, what I think is metallogorgia as well as chrysogorgia, at least 3 or 4 black corals in this area, species of black corals.
Okay.
Just call out if there's anything you'd like us to look at.
Anything we want to zoom in on just now before we pick up, Steve?
Yeah. I'm curious about that long stalk in the middle, the black coral with the what look like pink nodules on it.
Whoops.
Yeah, like granules.
Go ahead and push out in there, please, Jeff.
I'm wondering if those are animals on it.
Animal, mineral, vegetable. Do we need to get a little closer to see?
Yeah.
Probably.
Well,
I can bump forward if you guys want. I think that's as far as Jeff has on his zoom.
Yeah, that's full zoom.
this that's okay. Why don't we take a look around elsewhere first
Okay,
before we
sure.
make a move, just to make a plan.
Yeah, sure thing. Go ahead and come right there.
You can look over the spiky coral you were interested in.
Spiky.
The one on the far right?
Far right.
Over on the even further right.
Oh,
yeah.
The one over all the way over there?
I see, yeah. Maybe one of the ones in the foreground.
Okay.
There's these small bushy ones.
Like these?
Yes.
Okay.
Those are the one kind of at the very, very
I think it's
upper.
the same, but yeah, either.
Either one. Yeah, there's also that one right there.
This will get us some better light. Go ahead and push out in there, please.
Well, that one's a black coral.
Eyes like a
Black coral
hawk.
stauropathes. Okay. All good on that one.
Okay.
Good captures, and now maybe was one on the left-hand side that is a very.
This one?
Yeah, it's a branching fan to the left of that even.
Branching
right,
fan
Just to the
left.
right of the white sponge, yes, there you
Yeah,
go.
those right there.
Go ahead and push out in there, please.
It's another species of primnoid that's not over on the right-hand side.
Oh yeah, right. Can we get a little tighter on this one here?
Some bathypathes in the background. This one could be Narella or Calyptrophora. It's very difficult to make out, and the colony's not looking very healthy.
Yeah,
Missing
it
some
looks,
branches.
yeah.
All right. Thanks for that.
Yeah, sure. Go ahead.
Any others? There's some on the right-hand side, but I think it might be best to move over that
I
way.
agree. Yeah, let's pick up in lateral right just a little bit, please.
Yeah, sure thing.
While we're flying around, let's also be looking at this rock pavement, try to figure out what it is. Is it a sheet flow? Is it sediment? What is it? Crusted sediment. Yeah, a couple different things here.
Oh, that's a
Coming
weird
into view.
one.
Wow, yeah. That low one. Strange.
Yeah, there's definitely a lot of things we want to check out here.
I'm going to sit down here in front of these two, and you guys take a good look at these.
Yes, please. There's a couple things I think we'll want to look at.
I really want to sit on this guy. Sorry.
Jake, could we get the lasers on just briefly?
Yep.
Thank you.
All right, go ahead and push on in there, please, Jeff. Which one do you want to see first? Let's maybe go left.
Actually, both of these, yeah, the left and the right. This one, I'm not quite sure. It could be Narella, but it's tough to tell when the polyps are full out there. Although, actually,
Definitely a lot of attached organisms. A lot of brittle stars here.
Yeah, unclear, this one.
Okay. Would it help to look down at the base at all?
No, I was looking at the left-hand side branches.
Yeah,
It was
these over
actually
here.
really informative.
Yeah.
The right one, I'm more feel like I'm more confident with.
Okay, but
There.
let's still get a close
Is that a
up of that
crinoid
one.
on the right side?
Those left side ones, that was a different coral. I see a different base at the bottom.
Yeah, yeah, that might be.
Go ahead, Jeff.
At least maybe I'm mistaken. This is a weird one.
Yeah, so this one's actually fairly easy to identify. I think this is Narella macrocalyx. It's very large polyps. It's definitely in the genus Narella. We saw this earlier on dive Seamount A.
Okay.
The first dive, so
just a little further south of
just
here.
a bit, yeah, close by.
Yeah.
Yep. So that was
And what do we see
yeah,
in the background
one.
here, Steve? This, do we?
Oh yeah, there's a bathypathes in the
Yeah,
background too. Yep.
There's also a little yellow one right behind.
Yep, that might be Stichopathes.
The stick.
Yep.
Yeah. Oh,
Very
look at
nice.
this.
Lots. Another
Yeah.
little black coral tucked back there.
Oh, yeah.
Abundant.
Okay, yep.
Oh, yeah,
Great.
there's the Stichopathes.
Yep.
All right, we can pull out,
Pull
Jess.
wide. Sure thing.
Do we want to look at anything else while we're sitting right here?
We haven't looked at any sponges. Maybe we want to do a drive-by of the sponges on the way out.
Okay.
There's Rittigorgia in the background. There's also Hemicorallium in the background. So I think if we move a little bit or we shift a bit, we'll see a lot more diversity on the way out.
All right. Rennie, let's go ahead and call in that ship's move, please.
Sure thing. Jake, you're at 155?
155.
All right, I'll go 150.
Roger that.
Just follow that there. Bridge nav.
And if we could get some partial zooms of these sponges, that'd be great.
Yeah, sure thing.
Good evening. Let's step 50 meters, bearing 150.
Go do a partial
Speed
there.
0.2.
Do you want lasers on as we move?
We'll turn them on and off.
Thank you.
Thanks for checking. Wow, there's a lot of things attached to this.
We'll look in. Yeah.
Glomming on.
You come a little wide there. I'll go down to the bottom one here. Is this pink one the Hemicorallium, what we were seeing yesterday?
Yeah, I think it's probably
Can
very
we get the lasers
similar,
on, Jake?
if not the same species.
Go ahead and push on in there, please, Jeff.
Yeah.
It just might be growing a little bit higher up off the rock since the flow conditions might be different here, such that it needs to grow up a little bit higher before it has the same feeding capabilities as some of the corals on the other site, only because the flow conditions at the other site were quite good, uniform, and so things tended to grow fan-shaped right off the surface of the rock, whereas this one looks like it's
Got a
had
little
to
bit
grow
more
up
branch.
about 10 centimeters or so before it was able to feed and branch out.
Any other ones you want to zoom on?
Not yet.
Roger.
Bunch of Bathypathes and Chrysogorgia colonies, pretty characteristic of this depth.
A lot of brittle stars.
Yeah, that brittle stars, you'll see that when there's generally a little bit maybe higher productivity in the water column. There's more food particles, a lot more higher density of suspension feeders and filter feeders in the seafloor, all competing for that resource. Nice big Iridigorgia colony. I think I'm hoping and expecting to see a little bit increase in density as we move up to the top of this little high. There's still a bit of slope, so I'm going to hold off and requesting a water sample until we move up on top of this to see how it looks up there.
Yeah, and for those asking how tall these are, you can kind of get a scale reference with those green laser dots. Those are 10 centimeters apart. So some of these taller corals are well over 1/2 to 3/4 of a meter tall.
I don't know if you've already looked at this back row, but from Argus's sonar, we're kind of seeing a very linear ridge feature that we're on. It's somewhat flat like we're seeing.
Okay.
But we're not really seeing anything on either side, so it must drop off, which tracks with our map.
Does. Okay, thanks for
Great.
pointing that out, Rennie.
Yeah.
Beth, what is a sheet flow?
A sheet flow is a type of underwater volcanic eruption. So depending on the viscosity of the molten rock as it's coming out of the seafloor, if it is has a relatively low viscosity, then it flows very easily and kind of makes like a sheet as it moves down and cools. Whereas if you have a higher viscosity, or also sometimes if it has more gas in it, then you get kind of more explosive, eruptive, pillow basalt type features that look like kind of more like boulders. So it's not super easy to tell right now what it is we're looking at, but I'm sure as we keep going up, it'll become more clear. Is that this one here, Steve? Is that?
Umbellapathes?
Yeah, Umbellapathes is what it looks like. Here's a, can we have a partial zoom on this sponge to the right, please?
Yeah, sure.
Looks like a pitcher plant.
Kind of.
Really does. A little shrimp in front of it.
Oh,
Is it growing on the skeleton of the old sponge?
kind of. It looks like that.
Go ahead and push on in there, please. That's great. Thanks.
Can we get a little tighter shot on that? Thank you.
Sponges on sponges.
Yeah.
Yeah.
Actually, there might be another sponge on top of this sponge, so it's
Yeah.
sponges on sponges on sponges.
My goodness. I can certainly share, I suppose.
Thanks, Jess. You can
It's a sponge
in
fractal.
video. You can zoom out.
Mandelbrot sponge.
Yeah. There's some yellow fans here. These are probably Acanthogorgia. We can get one. Partial zoom, we can probably confirm when we have a chance. No rush on that. There's several around here.
Yeah, we can do it.
Some really big stalk debris around here. And you can see these large holdfasts attached.
Oh, yeah.
Old holdfasts. Can we get the lasers on briefly, Jake? Thank you. Yeah, so that lower holdfast is probably at least 20 centimeters across.
Jake, could you switch your sub-bottom range down a little bit?
Yes.
Make it a little easier.
And you can turn the lasers off when you are ready.
Coming
All
off.
right, go ahead there, Jeff.
Yeah, not sure about this one.
It looks very mucusy. Dense.
Might need to get a closer look at this one.
Okay.
Yeah, I mean, there are characteristics of this that suggest Acanthogorgia, but they look a little bit different than the Acanthogorgia we sampled earlier in the expedition. The axis is a different color, different texture, but the polyps are right. They're right density, yeah, right color. But with Acanthogorgia, you really can't be absolutely 100% certain until it closes up.
Okay.
So I'm hoping maybe we'll see one in the next several meters here that might have that condition. One thing I'm not seeing in this site, although maybe I missed it, are there any bamboo corals? They just haven't been here.
Yeah, we haven't seen, I don't think we've seen them.
Might be some dead bamboo coral skeletons, but I haven't seen any live.
A lot of diversity here.
Yeah.
There's 4 different sponges right there.
Yeah.
Poliopogon and Caulophacus both present here.
Oh, orange
There's a sea star.